Okay, top of the morning, everybody. Happy Wednesday. So, a couple quick things. I posted packback stuff on Moodle. There were some questions and concerns. I labeled it as week 3 because it was the third week of the semester, but it was truly the second packback assignment. Some people are like, what, did I miss one? No, I apologize. I changed the name on that if you have looked recently to week 2, because that's what it truly was. So all of that stuff got posted for that first bit because for lower division courses, ISU is utilizing a system called Navigate. You probably are familiar with that stuff, maybe. That basically is tracking stuff, and if there are issues with attendance or whatever, they potentially will reach out to you. But I don't take attendance, so how do I know? Packback was what I ultimately utilized. I mean, technically, the first exam could have been something that I used, but it closed at 5 on Monday, and they wanted the stuff Friday. I didn't submit stuff until Monday morning after I had packback stuff to look at. So that's what's up with that. If you did absolutely no packback, you likely got a message through Navigate saying lacking participation or something to that effect. If you saw nothing, then you've made some posts, all right? So any questions about that? Also, I still need to go back in there and manipulate. The total on there should be 60 points. Technically, there are a few that have done some extra, and there are some that have done less, all right? And I believe it defaulted it, set it out of 100. So you're thinking you just got a D if you did all of your stuff because you got a 60. But that's not really what's up. It's just 60 points. That's all I was looking at, all right? So I will try to manipulate that. But my other issue is when I drop it down to that, if you did other stuff, that potentially erases it off of there. It's not going to be gone. Don't worry about that. I will manipulate all of this stuff at the very end. I'm not going to take it straight off of Moodle. All right. So keep that in the back of your mind. If you see some stuff getting adjusted that way, I'm just trying to make it so that it looks like it truly should. Exam wise, all of that stuff looked great. I thought the average on it was 93, I believe, which I expected. It should be good. There were lots of good scores. It's open book and open note. You would kind of hope that you'd get a pretty good score. So if you got questions or concerns about that stuff, let me know. As far as some packback details, if you see your score on there and it wasn't the 60, you are welcome to submit extra posts during the current week, or you can hold off to another week, depending on how many you ultimately missed. The total amount is what I'm truly looking at for total amount of posts as I'm submitting those scores, not the curiosity score. So keep that in mind as well. All right. So we left off Friday about halfway through chapter 4, thinking about just all sorts of different organisms and the diversity truly of prokaryotes are what we're thinking about in this chapter. So we talked about Bacteroides and the potential for these to be some of the main colonists within the gut, providing essential nutrients, keeping a nice, happy gut. When we see things like proportions of some of our flora getting out of whack, there can be all sorts of issues that we might see. Clostridium difficile is a type of bacterium that can cause some pretty nasty GI issues. It's a bacterium that we have in our guts in super low proportions, held in check by a bunch of these other players like Bacteroides. And as long as you've got a plethora of these guys, it keeps some of the others that could be potential problems, keeps them kind of held in check. But if these guys get out of whack, all of the sudden stuff like Clostridium might say, sweet, look at all of this extra space, and it starts to proliferate, and then we start thinking about some ill effects. Sometimes it's through the use of some antibiotics, typically for something other than GI issues in the first place. You got something else going on. And as you take these antibiotics, it starts to impact other bacteria more than just the potential culprit. That may set the stage for the Clostridium to bloom, if you will. So thinking about this truly is important. And you probably maybe saw some stuff going through the pack back. When I was looking at this stuff on Monday, shortly after lecture, going through a bunch of things, I came across one post that was almost like right what I ended with. I was like, holy cow, this is awesome stuff. I believe the article came from BBC that they were citing. I read through that and I was just like, this is super cool. And I made some comments. So that article or that post should have been featured. So it should pop up and you can see it and potentially read through that stuff if you're interested in things like this. We said you are what you eat, and what if we are eating different things that potentially impacts all sorts of stuff? That article had all sorts of details about things like that. So pretty cool. All right, these Planktomycetes, these same players, two different scenarios with these guys. So they have the potential to tether or anchor themselves in their environment. Sessile means stationary or not moving. All right. So this holdfast is like an anchor, kind of gluing them right to where they would like to be. All right. Somewhere in the pond. All right. They have the potential to move. And the holdfast, we transition from this tethering and we can see a little flagellum over here. The swarmers, their flagella help to propel them away. Potentially, maybe in this circumstance, everything's fantastic. Ample nutrients, we're hanging out, pulling in stuff, growing. And then that starts to deplete, and we're like, holy cow, I'm starving up in here. I ought to move away. And in turn, they swim off with their flagella, hopefully finding an area more rich in nutrients and go back to the sessile habitat or environment that sets the stage for them to start being happy again, start growing. That's the idea with some of those. I think this is super cool, but just looking at it, it probably isn't something that you're like, wow, that is cool. It looks kind of just like a tube of grossness, right? This is called a Winogradsky column, and there are all sorts of cool things that we could think about or do with these. So Winogradsky was, I believe, a Russian scientist that microbiologist that was thinking environmental stuff, all sorts of different microbes in their habitat. How could we cultivate some of these things? How could we enhance what we've got just in the dirt and actually see it real time? All right. It takes time for them to kind of bloom, if you will, but you can watch these things change and different things start emerging and all sorts of microbial interactions in these columns. All right. So there's a lot of stuff that you potentially can do, but the idea is to provide some nutrients to the soil sample that some microbes potentially may like. There's lots of variation. You could set up a handful of different columns and use the same soil sample and then see how these columns emerge over the course of time. Give them a month or two, and you should start seeing truly some variation with it. So different carbon or nitrogen sources may be added to the media. Sometimes we rip up strips of paper, newspaper, or something like that to provide a source of nutrient. Other things could be added if we were curious about stuff like maybe bioremediation, and are there organisms that could be feeding on oil or feeding on heavy metals or whatever it might be. They could be added to this mixture and see what's truly available. So you basically take a tube, a column. You could use a 2-liter bottle if you wanted to do this in the leisure of your own home. You get something like some newspaper, rip it up, throw it in the bottom, dump some soil in there. Sometimes we take the soil and mix it with water. Sometimes you just add the soil and then water. I mean, there's multiple things. Usually a slurry. You play them with mud. We're making mud pies just like little kids, right? We never want to get away from that. So you mix the dirt with some water, make this slurry, and then you put it into your column. Ultimately, you're establishing an environment, a habitat in this column where different organisms, dependent upon where they're at in the column, have the potential to maybe grow, maybe thrive. Toward the surface, right at the surface, we're thinking more aerobic. And as you move deeper in there, an anaerobic sort of environment. We don't see the top of the column, but you keep a layer of water over the top of it. Now we're thinking like a pond, and you've got all of the soil in that pond and the water on the top of it, and all sorts of different potentials for interaction. Some microbes in the water, many in the soil, all right, and all sorts of variation. So we've got some arrows, super hard to see the purples, but there are some purple pigments that we can see. We call them purple sulfur and green sulfur bacteria. These bacteria, if you could use your imagination, you've got some purple or reddish pigments that the arrow's trying to point at in here, but it really, in this image, doesn't really do it much justice. All right. And then some green bacteria, some cyanobacteria. There's potentials dependent upon where your column's at to get other stuff like algae, like a fish tank that doesn't get cleaned very often, gets a bunch of algae growing on the side. You typically don't want a lot of that stuff in there. So when you make these columns, you try to put them in an area where they get like some indirect sunlight, a north-facing window instead of one that gets bombarded with light. And then it kind of minimizes the amount of algae that grows. You keep adding some water to the top of it as it depletes. And over the course of time, you can see all sorts of different things going on, all right? In microbiology, thinking environmental sort of stuff, these are common things to make in the lab. And you typically think about maybe early on, first bit of the semester, you make a Winogradsky column, and then over the course of the semester, you watch it toward the tail end. You've got a lot of stuff that you're able to visualize, make some samples, look at all sorts of different things, all right? And potentially something that just goes on for forever, all right? being a microbiologist, being a microbiology nerd, I built one of these with my oldest son when he was just super little, like 2. It is now 16 years old, and it just sets right down in my basement. I keep adding a little water, but there's all sorts of this stuff, purples and greens, all throughout, down toward the bottom, a little more anaerobic. All right. Super cool. All right. And sometimes when the water starts drying out, a little bit stinky. Hence living in the basement, right? So some more of our cyanos. Microcystis aeruginosa is a type of cyanobacteria, so an autotrophic microbe producing its own sugars. We can see these in all sorts of different freshwater habitats, and then potentials for a bloom, if you will. And we technically don't really want that, right? So with nice warm temperatures and also ample nutrients, as we see this kind of bloom of them, greenish water, it's butting right up against a bunch of agricultural fields. So we see runoff from the fields getting into the water. And this in ecology, we call this eutrophication, where you've got all sorts of microbes thriving in this environment. And first off, in the back of my mind, I'm like, sweet, look at all this food for these fish. But that's not what happens in these circumstances. You get these big blooms, and in these circumstances where microcystis is present, there are neurotoxins that the microbes start secreting out into the habitat. That's a detriment to the fish. There are other players that, as they're in there, even though they're photosynthesizing, they're metabolizing super rapidly when you give them all these nutrients. So they suck up a bunch of oxygen out of the water for their metabolic process, and it depletes the water of oxygen. They get massive growth, releasing toxins. It's a double-edged sword. And ultimately, the fish suffocate or get poisoned, or both. All right. Bad news. All right. So there's lots of push for where we see bodies of water and agricultural fields bumping up against them to try to buffer, thinking stuff maybe, although this isn't really wetland looking, but something like a wetland to trap some of that runoff before it actually makes it into the body of water to mitigate these issues. And we can see technically some examples of that right around American Falls with some farmers' fields that have transitioned some of the edges into wetlands. Pretty interesting. So a couple of interesting ones as we look at these Actinomyces israelii. These rod-shaped, but a little bit different as far as how they replicate, and they look kind of more like some alphabet soup, right? A bunch of Y's, branching sort of structures as they start replicating. So a little bit different. Corynebacterium diphtheriae, culprit for diphtheria. These bacteria, when they divide, it's sort of like how the actinomyces appear. They divide by the sort of, it's binary fission, but it's snapping. So it's toward the tail end, and they form these sort of V's, more alphabet soup. We call them palisades. And typically you see many in the process of this sort of division palisade arrangement. You've got a bunch of them in this image that are just hanging out, not in the process of replicating. So they're individual rods. But this one is, we caught right as it's starting to finish its replication, and they snap off like that. Gardnerella vaginalis. Here we can see some cells, eukaryotic, female pap smear is what we're looking at, I assume. Yep, there it is. So epithelial cells, and then some of the bacteria within there. Gardnerella is gram stain wise kind of an oddball. It's gram variable. So sometimes negative looking, sometimes positive, sometimes it's a little bit of both. All right. That causes a bit of confusion. And your first thought is, I messed up my gram stain, right? So you likely restain it and it shows up the same, but you were super careful the second time. Now you're like, aha, I think I know what I got going on now. If you're truly dealing with these samples in a clinic, you're going to know. All right. So a really pretty common culprit for vaginosis in women. We will go through some details end of the semester as we start thinking infectious disease, and we'll flip back to Gardnerella. As we start seeing some of these microbes, and this isn't the best image, typically the epithelial cells get just like covered. They look very different. You can see nuclei all over in here. And I got maybe a couple here and there, but usually they're like all over, and we call them clue cells, all right? Not the game Clue, but it makes me think of that, all right? Solving a mystery. So Clostridium difficile, we started with this today, thinking about GI stuff, all right? So I mentioned before, when it comes to thinking bacteria that we really want to focus on, Bacillus and Clostridium are the two types of spore-forming bacteria. Clostridium is an anaerobe, and the bacillus are aerobes. So different in as far as how they grow, all right, what they like, all right, but the potential for spore formation and the ability to persist and go on to survive another day is there, all right. Others besides our difficile, we think of as some kind of nasty situations, Clostridium botulinum or Clostridium tetani for botulism and tetanus. Bad news, all right? Clostridium difficile also can be bad news, all right? But this, like I said, something that we already got going on in our intestines in low levels, all right? When they grow out of control is when we really see the issues with colitis and diarrhea. You feel super, super bad, all right? And ultimately, different antibiotics typically get thrown into the mix to try to combat this. And there's other potentials. When we think about some of these infections, sometimes the patient gets, just completely, their flora, all of the good guys in there are almost completely wiped out. So as kind of a last ditch effort to help that patient, a fecal transplant is what happens. I think, poop sandwich, but that's not what you're truly getting. All right. It's going in the other way. Although I think, thinking some potentials for some pills that might, like probiotic, sort of an idea that might set the stage for maybe not needing the transplant. Ultimately, they give the patient good normal flora is really what we're trying to do. And this goes back, really, it's not the same, but it makes me think of that article that I was mentioning as we started this. When we think about our normal flora, everybody's got different stuff going on. We've got some definite similarities, and especially if we're thinking healthy situation, we've got some similarities, but proportions are going to be varied. There's definitely opportunities to see some different players in there. And there has been some evidence from these transplants that really makes me stop and wonder and flip back to kind of thinking article stuff about all sorts of diversity that we may have in our guts. And they've recognized individuals that need the fecal transplant could be overweight, and the person they got the transplant from was not, and the person receiving the transplant, the pounds just kind of drop off because the flora that they have received sets the stage for something different going on in that individual. And the complete opposite, a person that was overweight is the donor to a person that's not, and the pounds start getting added to that individual receiving the transplant. Crazy stuff. All right. A potential for dealing with weight loss, maybe. I think maybe a little bit crazy, but maybe it could be helpful. All right. Especially thinking like some serious obesity situations, maybe something like that could truly be a turnaround. It's hard to say. All right. Uh, but I feel like that's a catalyst for some pack back. It's got to be. That's super curious stuff, I think. All right. So then, that truly begs the question, how willing would you be to receive a fecal transplant? I think if you truly were on death's bed, you would be willing, right? But it's hard to say otherwise, right? So Streptococcus pyogenes is something that we all likely know a bit about and maybe all have had some experiences with. Strep throat is caused by Streptococcus pyogenes, right? This organism likes to live in our mouths, all right? You don't technically want it to be living there. Some of us could be carriers. Maybe you had multiple strep throats when you were a child. Maybe you got your tonsils out as a result of that. There are still potentials that some of these could be living there, but you don't see ill effects. You may have the potential to transmit it to someone else. Truly just kind of depends. All right. We will think more details, truly infection a bit later, and focus on some strep throat. If we gram stain them, they are gram-positive cocci, and they form these nice chains, as we see in this image, all linked together. Streptococcus pyogenes, if you've got a good culture of these, they typically show up like these beautiful chains and are really pretty easy to distinguish from other gram-positive cocci, all right? All of a sudden, you're just like, holy cow, there's chains all over the place, right? And it's a throat swab. I think I know what I'm looking at, right? So gram stain from either from this or from a throat swab could show us this. This plate that we're looking at next door is a blood agar plate. And Streptococcus pyogenes is a type of bacteria that we classify as beta hemolytic. There's 3 different categories of hemolysis. So can we use this red blood cell? Can we feed on it or not? All right. Beta hemolytic bacteria, like pyogenes up here, can fully break down those red blood cells. That's thinking sore throat stuff. That's painful, right? That's an issue. That's a problem as they potentially gain access and set the stage for other problems. All right. So this plate would look like dark red. You can't see through it. So opaque red, it's full of red blood cells. And where the bacteria is growing, it basically has cleared all of those red cells. Now it looks kind of like nutrient agar, virtually see-through. All right. If it has completely lysed those cells, beta hemolytic, you can see through the plate. Sometimes you hold it up and you're looking kind of at the light, and then you can see through it very well, just to kind of verify. All right. So all of these areas are where the red cells have been depleted. We started, this is some streaking for isolation. So we put our first streak full through there, flame the loop, cool it, do some more streaking, another quadrant, flame it and cool it a little more, and the last one, as we've spread it out, now we've got individual distinct colonies. We could do stuff with these, pick them off, transfer them, do whatever you would like this. And each of the colonies you can see kind of looks like a little halo around them. It's just a clearing of those red blood cells, all right? Beta hemolytic, all right? There are others that like the red blood cells, but they can't fully break them down. They partially break them down. We call those bacteria alpha hemolytic. And a cousin of Streptococcus pyogenes, Streptococcus pneumoniae, is alpha hemolytic. So it would partially break it down. Instead of being able to see through here really well, it's kind of brownish. It's broken it down, but it hasn't completely dissolved it all, if you will. All right. They look different, definitely look different. All right. So partial lysis, halfway cleared. All right. Partially degraded. Then the other, I told you there was three. The other type of hemolysis technically is no hemolysis at all. We call that gamma hemolysis. Gamma means we're just growing on the plate. It is just bright red, opaque, but there's colonies all over the top of it. All right. It can grow on it, but it can't use those red cells. It's using other nutrients in the media, not the red blood cells. All right. So truly, gamma and alpha are the breakdown, or excuse me, beta and alpha, and gamma, no use at all. All right. Pyogenes, beta hemolytic. All right. And don't think you're going to work with blood agar plates in the lab, but in a clinical situation, something that typically is done. If you go in for strep throat, and I assume a good chunk of you have gone to the doctor for thinking strep throat, you get swabbed, they do a rapid test, and they likely also culture it. All right. Sometimes when you go in there, you're maybe a little early on in the infection, and the ability to get a good amount of those and do the rapid test may show up as a negative, but when they cultivate it on a blood agar plate the following day, they'll see all of that hemolysis. That might be when you get a call from the clinic and they're like, we do need to give you antibiotics. You left thinking maybe you were good to go, right? But you were not, right? Streptococcus pyogenes definitely is an important one to think about treating with antibiotics, because it sets the stage for a handful of other ill effects as it potentially disseminates or causes other types of immune reactions as the result of just dealing with the infection. Trying to combat it is truly in your best interest. All right. There are a few things you might potentially employ at home. Staphylococci like salt, but the streptococci do not. So gargling with some salt water can create an environment in the back of your throat that's really pretty inhospitable to the streptococcus, and may potentially provide a little bit of relief, maybe setting the stage that you can drill them without the antibiotics. But typically, the odds are you ought to get some antibiotics, all right? Especially if a couple of times of gargling don't seem to really start doing anything. All right, shifting over. Bacillus, like I said, another gram-positive rod, but an aerobic one. And we can see it growing on this Petri dish over here. Bacillus cereus is a gram-positive rod that is an environmental microbe. We can see it on lots of things. It potentially can be found on rice. It can form a spore as that rice is harvested and it dries. The bacteria can sporulate the spores on the rice. Rice can get cooked and then not held at the proper temperatures and then consumed. and you can have some foodborne poisoning results as the bacteria has released some toxins in there. All right. They call it fried rice syndrome. There are issues for sure. You want to make sure foods are handled properly. So in this gram stain, we've got our bacillus, and we also have little bacilli, little rod-shaped bacteria that are gram negatives. So this is a mixture of Bacillus cereus and our good friend E. coli, all right? And you can definitely see some size differences besides cell wall differences. E. coli is very small compared to Bacillus cereus, all right? And just looking at E. coli, sometimes very difficult to really differentiate. Are these rod-shaped or are they cocci-shaped because of their super small size? This one's a pretty good one, though, showing definitely some rods with both. So this next image is a blood agar plate with the bacillus growing on it. So no E. coli up in the mix. However, the way that the bacilli typically grow could be an issue. If you did have this culture and spread it out, E. coli could be masked with the bacillus growing right over the top of them, all right? They grow kind of by swarming. They spread out. The bacillus have flagella. They want to move, and they grow really pretty rapidly. So if you did have a mixed culture, almost always the bacillus just kind of covers them. And if you grabbed what you thought was a pure colony right here and stained it up, if it was a mixture, you might see something like this, all right? So seeing growth like this spreading out, really sort of difficult to get some individual colonies just because of how that happens. Kind of a giveaway that it's a type of bacillus, maybe not necessarily serious, but likely one of the bacilli. And if your gram stain showed a positive rod and you see growth like that, that's what I'd start thinking. So this image is Staph aureus. This is not a gram stain, an electron micrograph of it through a gram stain. And we showed one before where they colored it purple, and it looks like a bunch of purple grapes. Staphylococcus is gram positive, all right? This one has been colored differently, and it looks more golden, all right? And I kind of like that. Now we're looking at some golden grapes, right? Green grapes, right? These golden grape clusters that we're looking at here, I like it because aureus means golden. And here we've got it, all right? When you see not through an artificially colored electron micrograph, but when you see these growing on a plate, oftentimes the colonies have kind of a yellowish or golden sort of pigment to them, all right? Not a dead giveaway, but sort of, all right? Staphylococcus epidermidis doesn't produce that. It's more of just a straight white, very small colony growing on those plates compared to aureus, all right? So, like I said, kind of like that they made it golden so I could spout off that little detail, all right? Every time I see this, I think of cauliflower because that's what it looks like to me. And it looks like cauliflower that has been slathered in butter, so you know it's going to taste good. You douse it with butter, it's got to be good, all right? So that's what I imagined when I see it, but that's not what I'm looking at. So this is Mycobacterium tuberculosis. It's growing in this case on a specialized type of media called Lowenstein-Jensen agar. It helps to enhance the growth of our mycobacteria and also inhibits a bunch of other cells from being able to grow. And this is super important because when it comes to trying to cultivate tuberculosis, these guys take forever to actually grow, all right? To get visible colonies to show up. Patience is needed, all right? We roll into the lab, we inoculate something, and you roll in 2 days later and see your results. Technically, you could roll in the next day and see your results. All right. Very quick to get growth on the media. Some stuff takes a little longer. So those environmental ones took a little bit longer. But most of the bacteria we work with, super quick. All right. These can take weeks to double from one cell to two. All right. To actually start seeing stuff in a culture, in a tube or a plate where they're trying to grow, might take almost a month before you've got actual stuff you can work with. That's a huge issue. All right. So we see sort of the shiny looking growth on here. Sometimes you see really shiny growth and it looks slimy, kind of snotty. All right. This growth indicates to me that what I'm looking at are some bacteria that can produce a capsule layer, a sugary coating, and it becomes really kind of mucoid sort of colonies. This looks different, right? It's shiny, but it's not the same. It's not snot looking, all right? It's not mucoid, all right? It's different, all right? Uh, Buttery, right? And this type of image that we're seeing in here is because these bacteria have the mycolic acid in their cell walls, wax-like structures. Waxes are lipids, butter, right? But it's not a buttery coating, but it's kind of the same idea. All right. So this mycolic acid that these cells have in their cell walls gives them sort of this glistening sort of appearance as we're looking at them on the plate. All right. We can do an acid-fast stain and detect acid-fast bacilli straight out of the culture, the sputum sample from the patient. That's the only staining procedure that we can get a positive identification, see acid-fast bacilli from this patient, and say with certainty, this is tuberculosis. And we start treating with antibiotics immediately, and then we cultivate this thing. And a month later, after we've got good culture, we've done antibiotic tests, and then we potentially go back to the patient and we change the drug, all right? Because maybe they are resistant to what we were treating with. Maybe we need to alter the dosage. There's all sorts of differences. We don't get that till after we've already been taking these antibiotics because you got to grow it up to figure out some of those details. Kind of crazy stuff, right? And like I said, it's the only one that we recognize through that specialized staining procedure and immediately know this is who we're looking at. This is what's up. You've got TB, right? Done deal. Terrifying. You don't want to hear that, right? And many thinking going into health professions have had or will have TB testing done to make sure that you're good to go, right? We'll see some of that a little bit down the road. How many people have already been TB tested? A good chunk of you. All right. I bet there's some Zoomers that are in the same boat. All right. So as far as our phylogeny goes, we've seen this stuff before, a little bit of detail with that as we talked about separating our prokaryotes into the bacteria and the archaea, talking a little bit about it. All right. So what we're thinking about as we look at this figure is this acetothermos, this little star clear down here at the bottom of our tree. As we get to this bottom bit, Luca, the last universal common ancestor, bacterial-like, and we see speciation event giving rise to the bacteria, all right? And then differentiating to archaea and eukaryotes as we march our way up through this tree. We want to look at this little branch clear down here at the bottom, all right? Actinothermus, we call this a deeply branching bacteria. It's coming right off toward the bottom. and we don't see other little things coming off of it. This is a super ancient type of microbe bearing similarities to what we envision were like Luca, all right? And knowing details about this allows us to make some speculations about some of this as we're moving down here, all right? What sorts of things were they able to utilize and definitely not utilize, like oxygen, all right? All sorts of different potentials. And with thermos in the name, heat lover, all right? And also probably with the aceto, I envision acid, all right? So tolerating low pH, high heat, deep in the bottom of the ocean, hydrothermal vent sort of stuff as a potential for some of these microbes. Super interesting, all right? And then seeing differentiation as we march forward to some that we will think about as we progress through here in some pretty good detail, all right? So we'll get to more. This, every time I see this, I'm just like, man, so crazy. Dinococcus radiodurans, you know Conan the Barbarian. They call this Conan the Bacterium, right? This sucker can live and survive through all sorts of stuff. The strongest of them all, Conan, right? Crazy, all right? Dinococcus can thrive in habitats that we would not even think of having the capability of surviving, all right? They can live in the cooling ponds of nuclear reactors, all right? Where all of the fuel rods are, where if you were to even probably step into this room without proper clothing, you're not stepping out of it, all right? And they're living, thriving, all right? Feeding on stuff, all right? That makes us think astrobiology sort of stuff. Could these be microbes that we might potentially find someplace else? Maybe you've seen some details that the potential for life on Mars is maybe something that we're on the cusp of saying it's there, right? There's microbes down there, right? Are they like this? Are they different? What can they use? What can't they use? All sorts of ideas when we start thinking about some of that. And Dinococcus gives us the potential to extrapolate some of those thoughts outside of the Earth, all right? There are lots of people interested in Dinococcus, all right? Department of Energy, curious. Department of Defense, curious. What the hell's eating all of this radioactive stuff, right? We would like to know, right? That's a potential issue, right? Maybe interesting, all right? So sidebar briefly, all right? At ISU, we've got all sorts of different types of research happening. The physics department has a linear accelerator. It is nowhere near lower campus. It's up toward the going up the hill in a building that doesn't really almost look like a building. You kind of see this structure that looks sort of like that, and the rest of it is in the mountain, hidden away, sort of, all right, and contained, all right, super high energy. So this linear accelerator gets electrons super excited and shoots a beam of electrons at something, all right? And they are using this to produce all sorts of different things, isotopes for medicine, lots of different things. There was a researcher here in biology, not working with Dinococcus, but it was from observations of it that set the stage for her research. And some really pretty interesting stuff. Dinococcus can communicate with other microbes, and it can protect itself against high levels of radiation, and it can tell others about it and say, ramp up some protection. We call it some bystander effect, where you're telling your neighbor and whatever, and a leg up to persist. So there are other things like salt-loving organisms, halobacter, which is technically an archaea. That's what they were working with. And it's salt can be an adverse habitat as well, not as adverse as radiation, right? But they were bombarding electrons at halobacterium and then plating it out to see, is there some bystander effect? Are cells that were hit with this high level of radiation impacted? And how are they? Do they tell their neighbors about it? And do the neighbors ramp up protective mechanisms? Super cool stuff. All right. And collaborating, most people in biology aren't thinking, I ought to hang out with a physicist, right? I know going back to my undergrad, physics was a bad word, right? That class was not fun for me, right? But going to the linear accelerator and seeing what was going on, I'm like, I'll hang out with these physicists to see this stuff happening. That's cool stuff. All right. All right. Enough of that. I will quit there. Farewell. Good day.